Okay, so today we will continue to discuss the examples, the key examples on the biophysics, especially the physical technologies, physical tools that the people use to study biology and then get a new insight of biology. And then those new insights can be very useful, can be very transformative to advance our understanding of both normal physiology as well as the passive biology for the different diseases. Okay, so then this can be helpful in our human health care, right? So before I talk about those interesting examples, two new announcements. First one is I uploaded a book that's called Basis of Life onto the canvas, so that you can see many chapters in the book. So that book can be used as a reference, and you can start to read that, because in the next few lectures, we will talk about the 4 different building blocks of the life of the cell. And this book actually contains very comprehensive information. So anything we discussed in the class will be overarching the most key point, but the detailed materials, many of them can be found in this book. I have several other books, very useful cell and molecular biology books. I will also upload them, but you can read this one first, okay? Second announcement is next Monday, we will not have class because of a Labor Day. So I will see you Wednesday then, okay? Okay, other announcements are the same as previously, and some of you actually send me very good survey. So far it seems you guys are doing pretty well. So I'm glad that you are learning. So good. So keep sending me the survey, and then remember the questions I asked. What are you confused about? What do you want to learn more? What do you feel interesting you learned in the class? And also what other changes you hope to make in our class, right? So feel free to send me those opinions, and if you have new comments, feel free to add into them. Okay, that would be great. This will benefit all the students. Okay, yeah. Okay, so let's start our today's materials. So in last lecture, we talked about the definition of biophysics, right? So this biophysics is the field that applies the theory, mostly is a quantitative physical theory. This physical theory contains physics, mechanics, and also mathematics theory, actually, okay? This theory is very comprehensive. The physics theory contains optics, electromagnetic theory, and also thermal theory, because all those different physics existing, those different physics derived, different physics theory derived from the macro world also exist in the micro and the nano world. Okay, so the laws there are still applicable to the cell and also the subcellular regions, and also the methods of the physics. Okay, different technology of the physics, use them to understand how biological systems work. Okay, and then we give the examples, what kind of question, what kind of biological system questions people are interested in. Okay, here is just some examples, but there are many more questions there. Okay, people use the physics theory and physics method to study. Okay, and then we said we already now learned biophysics is a field that talk about the quantitative science. So all the description there actually is quantitative, not just the qualitative. Okay, we also talking about the subtle difference between biophysics and the biological physics, right? And then for the biophysics, they use those tools to understand the biological system and then learn new biology, means get new insights of biology that previously is inaccessible, previous unknown. Okay, so At that time, Daniel asked very good questions about what does this mean. So I explained it in last class. In today's class, we will talk more about this, okay? Throughout the semester, we actually will study both biophysics and then biological physics, both. And then we will emphasize on getting new biology by applying the new physics theory and physics tools, such as the microscope, laser, X-ray, and nanotechnologies, okay? We also discussed the analogy that two terms looks quite similar. Things are talking about similar things in the field, but they have a subtle difference there, okay? So such as biomechanics versus mechanobiology, okay? Can you guys hear me? Can you guys hear me well?
Yes.
Yeah.
Excellent, excellent, good. So now let's go back to look at the biophysics. So biophysics, let me close this for now, move this away for now. Okay. So biophysics is using the physics theory, physics tools to study the biology. And then the interesting biology, interesting and meaningful biology that people like to study, actually including force, energy, structure, and dynamics. Here, this force is not only mechanical force. So you have, if you learn the fundamental mechanics before, the Newton's law, right? You have a force applied to the body, and then body can move, and then what body will acquire the acceleration, right? This is a mechanical force. But this force is not only that. This force can also contain chemical force. So if you learned the thermodynamics before, when chemicals have different gradients, they will have different chemical potential. And then the components, elements, ions, they can move, right? So that's called chemical force, okay? This energy is also not kinetic energy and potential energy that we learned in the dynamics class, okay? This energy also contains electrical energy, magnetic energy, okay? So it's a very generic term, energy. Structure, what does it mean? Structure. Structure means the layout of different components of the cell, because this structure will determine the function, and the function will reshape the structure. So those structure is very important to look into. If for example, if neuron has disease, such as Alzheimer's disease, people tend to forget things. At that time, to detect Alzheimer's disease, we should not wait until people show that symptom. If people start to forget things, then you detect them based on the factor. People are forgetting things. You say they diagnose the patients as having the Alzheimer. That's too late. If people detect the neuron before patients show the symptom, showing the neurons connection start to be weakened, or the neurons membrane shows many plaques, shows many agglomerates there. That's actually the structure change of the neuron. That's a signature of Alzheimer's disease in a term of 10 to 20 years. Okay, if the neuron show that symptom, show that kind of structure change, then that means after 10 to 20 years, the patient will develop Alzheimer's. So then, if we can see those structures earlier, we can treat the patient earlier to prevent the further development of the disease. Okay, and then dynamics. Finally, is dynamics. This dynamics actually contains all these important parameter, force, energy, and also the structure change. Okay, so dynamics contains all of that. So it's actually a functional change inside. So all those information are very useful, and then they can be studied by physical tools. Okay, here on the left of the slides, we show several traditional tools. Those tools all have been developed for a very long time. Okay, so they have been used to study the biology at a different skills. First one is called AFM. This actually stands for atomic force microscope. So this device is actually really small. This is a picture taken by the scanning electron microscope, SEM. Okay, so it's not using the optical microscope; it's electrical microscope. So usually electron microscope look at the sample at a very small size. It's smaller or close to the light wavelengths. Okay, so that's why the light, if you use light or microscope to look at them, the light can diffract, and then it will blur the view. But the electrons, they have much smaller wavelengths compared to the photon, right? So that we use the electron to look at the small objects, then we can get a very sharp visualization. So this AFM actually is the sub-micrometer scale. Okay, the contents are cantilever. This is actually do you guys see my mouse? Do you see my mouse?
Yeah.
Yes. Yeah.
Okay. So the AFM contents are cantilever. This is a cantilever, very soft cantilever. And then at the end of the cantilever, there's a tip. So this is a perimeter. Inverse the perimeter is actually a tip. Okay. And on top, this horizontal one is the cantilever. This cantilever can be bended. Okay. Whenever it's bended, the deflection of the cantilever can be monitored by a laser that is shining at the back of the cantilever. So any small deflection of this cantilever, up and down, okay, up and down, can be detected. That can reflect how much force this cantilever is experiencing. Okay, and this tip is used to touch the sample. The cell or the molecule is at the bottom. Okay, so this tip is used to shake the hands with the cell. Okay, and then it go vertically, indent the cell, inwards, downwards to indent the cell, or go upwards to stretch the cell. And during that time, cell shape can be monitored, and then the force applied to resulting the change in the cell shape can be measured by the laser on top of AFM. Okay, they can measure the deflection of AFM. So this tool is actually very useful. It's very useful. It has been used by people to measure single molecule deformation. So single molecule has a single molecule has a small force between the bonds. Okay, and then those single bonds can be measured by the AFM. Okay, so this is a very useful tool. Another one is called X-ray. So this X-ray, actually, we saw this picture before. This is the DNA structure in the Fourier plan. So I mentioned that a scientist, a lady, right? She discovered the symmetrical structure of the DNA using the X-ray method. So this X-ray, you see, also this is not the light, right? It's not the photon. It actually has it has the smaller wavelengths. X-ray has smaller wavelengths than the light wavelengths. That's why it can be used to look at the sub-nanoscale molecules and then find out the light, the X-ray scattering patterns. Okay, so this is a very useful method to do the molecular imaging. The third one is called NMR, nuclear magnetic resonance. This is always used to check the patient's body, the molecules components, the element constitutions in the patients, because the mechanism is this is a very useful physical tool. This is a Nobel Prize as well. Optical trap, the fourth one, optical trap, is also Nobel Prize. They both are Nobel Prize. The theory of X-ray is also Nobel Prize. AFM actually yield multiple Nobel Prize. The tools. Okay, so for the number three, NMR, the theory is it applies a magnetic field to the patients or to the cells, because because every atom in the cell, they contain the nucleus. Okay, every atom contains a nucleus, and then surrounded by the electrons. And then because nucleus has the charge, positive charge, and then by applying a magnetic force, by applying this magnetic field to the cell, every atom inside their nucleus can be rendered with the energy. You can use this magnetic field to give energy to every atomic nucleus. And then that energy can be released by those nucleus as well. That released energy is associated with a wavelength. That wavelength can be captured by the MMR device. And then using that reading, people can draw a spectroscopy diagram. Okay, as the peak, the energy versus wavelength diagram. Different molecule, different atom, they will have different energy gain and energy loss, which will yield different wavelengths and a different intensity. From that, we can tell what kind of molecular composition in the cell, what kind of molecular disease in the cell. Okay, this can be detected. So MMR is a very useful way to look at the molecular composition as well as molecular structure. Okay. Number 4 is the optical trap. So this is a very delicate method. It's actually complementary to the atomic force microscope. It also can directly apply force to the molecule and to the cell, but at a smaller range compared to the FM. So in this optical trap, you actually put a particle, transparent particle there, and then let the light go through the particle. When the light goes through the particle, light will bend because the reflection index of the particle is different from that in the air. Then the light will deflect. The deflection of light actually introduce a moment mismatching. Moment mismatching will result in a force, mechanical force, that push the beads towards a certain direction. So that means we can use the light to apply the force to the bead and then move this bead. So scientists actually bound this bead to the cell or to the molecule and then push the bead away so that we can stretch the molecule and stretch the cell. Okay, so that's why it's called a trap. And we use the light to control this bead. So that's one called an optical trap. Okay, optical trap. Sometimes people call it optical tweezers. Okay. Can you guys hear me?
Yep.
Yeah. Okay, very good. So these are very typical physical tools people developed using the physical laws to understand biological systems. Okay. About 10 to 20 years ago, there's also the advances in the imaging field. So that's one important method called super resolution imaging. Super resolution. So this is actually using the light optical microscope. It's different from X-ray. X-ray is using not using the visible light. Super resolution microscope is using the visible light, but it can go to a much finer resolution in contrast to normal optical microscope. Okay, normal optical microscope, the resolution is a half wavelength. Okay, the wavelength is visible light. So usually visible light is 400 nanometer to 800 nanometer, right? From the blue to the red, 400 to the 800. And if half wavelength, if it's in the middle, it's about 500, 400 to 800, about 500 nanometer is the middle. And the resolution is half wavelength, then it's 500 divided by 2, 250 nanometer wavelength. So then that means any object, if they are smaller than 250 nanometer, you will very hard to see it. And if there are two objects, they are close by, and they are the distance is less than 250 nanometer, then in the optical microscope, you will view it as one object. You will not be able to tell there are two objects there. Okay, so that's a barrier for us, because many things, many molecules in the cell, their size is less than 100 of nanometer. So if the normal optical microscope has this restriction, they can only look at the things or only distinguish objects larger than 250 nanometer. Then that means many other information were lost. We don't have that. Even we take a picture, the information cannot be extracted out from the picture. So to address that challenge, people developed the super resolution microscope. I will discuss about the mechanism in the next, when we get to the section, talk about the imaging, okay? But overall, using the super resolution imaging, the resolution can get to 10 nanometer, okay? So it's from 250 nanometer to 10 nanometer, 25 times finer resolution, okay? Using that, you can see many new things. So here is one example. What you see here, this is a neuron axon, okay? Every neuron has a cell body, and then have a long axon. That long axon will connect the neuron to their neighbors and then transmit information. This picture is in the super resolution microscope. Then you can see, and here you see the fluorescence. This is actually the staining of the actin. Now, using a super resolution microscope, people find there are this axon, okay? They find perpendicular to the axon's elongation, there are many lines there, many lines, periodically, many lines there, okay? The line to line distance, I believe, is 7 to 10 nanometer, 7 to 10 nanometer, okay? 7 to 10 nanometer. So many of these lines, okay? And then here is the magnified view, okay? This 7 to 10 nanometer is exactly the resolution limit that can be viewed by the super resolution imaging. Previously, using the conventional microscope, we don't see that. Only by super resolution imaging, we can see this periodical structure there. And then those structure, later on, people find this structure is used to sustain the geometry stability of the neuron. Okay, without this structure, axon is very floppy, very flexible, too flexible to sustain the neuron shape. Okay, this periodic structure is like the multi-layered reinforcement of the geometry to let the neuron be stably and forcefully stay in the place they should stay. Okay, so this is a kind of new biology I was talking about. New biology, using the new physical tools, you get new biology. Okay, and then those new biology will tell people new information, and then it will be very interesting to see the neurons in the patient body, like Alzheimer's disease or Parkinson's disease, how those structure change. If they are changed, can we restore those periodic structure to help a neuron to gain healthy functions, right? So those are actually the meaning of the fundamental science here, okay? So this is structure, okay? So the first some tools, they are used to measure force, also can measure energy. And this tool, super resolution imaging, measures the structure, okay? So last one is dynamics. So here is one video. Can you guys see the animation? Can you see the video here?
Yeah.
Yeah. This actually is generated in my lab. This is real data. Okay. So here what you see is a bunch of cancer cells on the 2D substrate. Those are colon cancer cells. And you can see those cancer cells, actually, they can generate red color, those waves. You see, this cell develop the wave here, this spot and this spot. They develop a wave. And then this wave will spread away, right? This is the dynamics. So it shows that a cancer cell, they are not static in their own location. They actually actively communicate, talking to the neighboring cell that is even they are far away. Okay, and then guide the other cells to perform certain functions. We have found that those waves can guide the cells as many malignant functions. Okay, so this is not the structure information. This is dynamic functional information. Okay, and using those using the new imaging modality called functional imaging modality, then you can see the functional dynamics inside the cells. Okay, so and those research are still ongoing. There are many interesting things currently we find about the unique physics in those cells. Beyond that, using the physical tools, we can also look at the even larger view of the biological system. What I just showed you in previous slides is cells, single cells, and the cell clusters. Here in the video, can you see the video? Can you guys see the video?
Yeah.
Excellent. Here in this video, this is a new microscope system. Using this, you see a whole organism. Okay, this is a small embryo of a fruit fly, small embryo. The color, you see the fluorescence, actually color, is the nucleus of every cell. What you see here is the 3D view. It's actually 3D view of a small embryo when it is just yield. And then how the small embryo gradually develop and then develop into the larva. Okay, and then from the larva, I didn't put another video here. It's from larva gradually develop into the whole animal. Okay, and then the fly will really fly away. Okay, so this is a new microscope system. It's a 3D system. Actually, in my lab, we have this system. We use that to image the 3D neuron communication in the brain of a larva zebrafish, and then to tell how the zebrafish think when they experiencing the external stimuli. Okay, now that's the ending of the video. So here you see at this stage, the larva start to have a dramatic movement here. Okay, and then this is a start point. Okay, so this microscope, those microscope and all those imaging methods can help people to understand the living system to a much more deeper manner. Okay, so this indeed is a data-rich science, right? Data-rich quantitative science. Yeah. Okay. So beyond that, all what I'm talking about are the different physical tools. Of course, there are also physical theories very important. So in the biophysics, people also use very useful knowledges to do the modeling. Okay, so here is one example about the metabolism inside the cell. Then we can use different physical theories and the chemical kinetics series to and also electronic series to describe different parameters in the cell during different processes, and then quantitatively lay down the governing equations, and then use those equations to derive a unified framework to describe cellular process in the cell. Okay, and then those equations, we can verify them using the imaging tools in the experiment to prove this is true, and also to predict, use those equations to predict new phenomena that have not happened in the experiment, but it will happen during certain circumstances. Okay. So in this biophysics field, it is actually very significant, very useful to understand how to integrate biochemistry with the force field. Okay, and then this force field will include both mechanical force as well as other factors, including the different boundary, okay, of the cell, and different interface of the cell, cell contact, and also the flow in the cell. In the cell, they are actually the fluids are moving; they are not static there. And then also the electrical field in the cell, they also have an electric field. Okay, and then during what I'm saying here, these are governing equation based on those different conditions, and different force field. Then we should decide which parameter should be included in the model, which one should not be included in this model, and then generate the final integrated model to describe the biological process. Okay, so this is a very, very useful way to understand the living system. Okay. Okay, those are brief introductions about the biophysics field using both experiment tools and the physical theories to understand the biology. Now, with that, if you master those tools and theory, then the next question is what kind of biological question you want to look into, right? So then we need to know what is the current challenge, what is the most important question we should study at the given time. Okay, so we know that life is driven by the numerous micromolecular machines and the cells. Here is one example showing the kinesin motor protein working on the microtubule cytoskeleton, right, carrying vesicles. Now, in the body, we have billions of those motor protein, as well as other types of motor proteins. Now, with that, what kind of things we want to understand better? Here is a list of the interesting and important questions that people like to address. Okay, and those are the ground challenges currently in the field. Each ground challenge, if we can address that, then the science will step forward will go forward for a big step. Okay, that's huge. So first question is, how do they work in real time? It is not known yet. How can all those motor protein or other molecular machine work in a real time? Right now, this is not known. Currently, people only take a single cell or piece of some motor protein, some concentrated motor protein as the molecule onto the glass cover slide, and then watch how they move individually. But how they work in real time, especially in the body, in vivo, in the real body, how they work, that is not known. Because imaging tools is not going deeper enough into the tissue. Okay, and also how do these molecules work either individually or together? There are still many mysteries there. That's not known. How they work inside the living cell. This actually is 3 questions. Actually, is talking about from a different aspect, but altogether they are talking about how the different molecules communicate with each other and coordinate together to perform the biological function. Okay, so these are not known yet. Okay, also a very interesting question is how do the genetic information, we discussed about genetic information, right? They are stored in the nucleus. How do those DNA sequencing determine the cell function? We know the DNA will give rise to the RNA and protein, but then how do different DNA, how different gene give different proteins, and then they coordinate together to realize certain functions, certain cell functions. This question also is to be addressed. Okay, and finally, how do cells? We have different type of cells: endothelial cells, fibroblast, muscle cell, neurons, and also cells also have different states. Okay, some cells are in the happy state, some are in the angry state, right? Just like human beings, they have different modes. Cells have different modes, and also cells have perform different functions. Those are very complicated combination. How do those combinations work together to form the tissue and form the organ and form the whole organism? Okay, how do this happen? Those are actually bigger questions. Those are very big questions. Every time when you do a research project, you may address just one piece of those questions, but you need to make sure that the question you ask, you work on in that project is the answering the important part of this, can move our understanding forward to address those questions much better. Okay, so you want to pick up the story, pick up the project like that. Okay, good. Now those are the examples I hope to share with you. Now, next, we are going to talk about the necessary biology, necessary physics laws we need to know so that we can use them to do the research in the lab. Okay. First of all, I hope to introduce some basic molecular and cell biology knowledge to all of you, because some of you may not know, may not have training in the biology, especially in the quantitative biology. So I want to keep all of us on the same page, and I will be brief, and I will capture the key point for you. Okay, it will be very useful that you get yourself familiar with the quantitative ways to think about the biology, not just take a normal biology class. Okay, so to quantitatively think about the biology, we need to think it like a physicist, like an engineer. Okay, think about it that way. So yeah, here is the thing I hope you to master. Firstly, we need to understand the biology using some simple ideas from physics. Usually, the traditional biologists, they will understand biology using quite complicated ideas from the biology. There are many terminologies, many different names that biologists need to memorize, and then they use that to understand biology. I think that's very important for them and for all of us to know biology, but that's not sufficient. We also want to understand the biology in a way that it can help us to highlight the important part, the key part in the biology, away from the travel part of the biology. Then we can focus our limited energy and resources to understand the important part first. Okay, so I think that's the different, that's the compatible ways to understand biology. In this class, we will use the simple ideas from the physics to understand the biology. Okay, and then of course we need to know enough biology so that using the traditional way to know biology, so that we have a sense of what to study. Okay, and I don't assume you have any biology on the knowledge. Okay, so we will introduce everything we need in the class. So here, firstly, I want to ask you a question. If we want to understand the biology from using the simple idea from the physics, very fundamental hallmark of physics is there are many equations, right? Every field in the physics has an equation, have the law, and that law is represented by the mathematical equations. Now I want to ask you a question. That is, does biology also have laws, series, or the equations? Do you understand what I'm talking about?
Yeah.
Yeah, I think so.
If you understand it, can you tell me, does biology also have laws, series of equations? Can you tell me?
Like the theory of evolution?
Theory of evolution. Very good. That's a fundamental law developed by the Darwin, right? That's one. Excellent. Any other law, but that one doesn't have equation, right?
Yeah.
Yeah. Tell me other laws, and also especially the equation. Is there any equation?
I think
Could you apply conservation of energy to the system, a biological system?
Conservation of energy into the biological system. Can you be more specific? As we know that all the physical laws may be applicable to the biology samples, because the biology sample is also the entity in the universe. So physical law applies there. But I'm not asking that. I'm asking that the biological behavior can the biological behavior be described by the new equation? That equation is not previous equation. We are not applying previous no equation to the biological entity. I'm saying that biological entity behavior can they be described into the new equation? You understand what I'm talking about? Do you understand what I'm saying?
Yes, so something more specific to a biological system.
Yes, yes.
Like
not the
metabolism,
the
maybe?
equation of physics equation that you apply there. Yeah, good. Go ahead.
Oh, I was gonna say maybe metabolism, because I mean there's some biochemistry there.
Excellent. The excellent. But as I showed in the previous slides, many metabolism, many equations that are used in the chemical engineering that you can write that equation and then use that to describe a certain process, right? But that can you give me? But that process, that equation is from chemical engineering. I wanted to apply that to the biology, and then go beyond the equation coming from the chemical engineering books.
It's not quite an equation, but because a lot of the equations that have popped into my head are more rooted in either physics or chemistry,
Good.
the closest thing I can think of that is specific to biology is if you described a particular biological pathway, like I'm going to use the hippo pathway as an example. If you could describe that as a sum of components to reach a particular end result.
You're talking about pathway. Yeah, pathway is useful. Sure.
One equation that I thought of was when you were learning about like a food pyramid and how much energy transferred to the next trophic level. It's like 10%. It's a very simple equation, but it's usually used in biology, I guess.
That's a still similar thing as Jackson talked about. You are talking about some equation that already exists in the other field, and then you apply it to the biological samples, and then you said this equation is a biological equation. That's not what I'm looking for. I'm looking for in the biological field it alone, there is an equation there. You know what I'm talking about?
Law of segregation, you mean?
Law of what?
Segregation?
Migration?
Segregation. I mean something genetic, gen?
Are you saying law of segregation?
Oh yeah, same.
Law of segregation. Any other things?
I think he also said like genetics.
Excellent. That genetic that developed by the Mendel, right? Mendel. Is that what I'm talking about? Genetics.
Yes.
That's excellent. That's actually is a law, not an equation. That is or theory. We can call it a theory, right? Yeah. Anything else? That's excellent. You guys are really excellent. Anything else? Okay. Good, good. This actually you can keep thinking about this. This is a very, very interesting question because right now, as the research goes, people actually are looking for the rules of the life. So previously, the life, people think that there's no rule. For biology, there's very few rules. What you said, two are very important. One is evolution. Another one is genetic theory. Okay, these two are very important. But beyond that, is there any other rules governing the life? So I'm not looking at just one cell. So if, for example, what I'm talking about is in the lab, you are working on the endothelial cell. You find some behavior there. Can you extract some laws, theory, or equation from the endothelial cell? And then that is also applicable to the muscle cell, also applicable to the neuron. Can you do that? That is the governing rules of life. That is some things people are actively working on. Okay. Good discussion. Good discussion. Now, here, talking about the laws, here are the five physicists who develop laws in the physics. Okay. So before I go to the laws in the biology, I want to ask you whether you are familiar with this. Do you know who is this person? And can you tell me who is this person and what kind of physics law or equations or theories he developed? Can you tell me?
I believe
Newton.
I think it's Newton, right?
Yeah, Newton and the three fundamental laws of physics.
Excellent.
well, the three laws of motion.
Excellent. Newton. Second one. Second one is Maxwell. He developed the Maxwell's four equations: electricity and magnetism. Okay. Fourth one. Do you know who is first one? Third one. Third one. Okay. Gibson. Okay. He developed the thermodynamics, and there's an equation about the Gibson's free energy. Okay. Number four is Boltzmann. Okay. He developed the statistical mechanics of entropy. And here is the equation right on his tomb. Okay. Equation. Who is the number five? This person, number five, actually is the person most close to the biophysics. You see the first four, they actually is a traditional physicist. They don't work on the biophysics, even though the laws or equations they developed can be applied to the biological system. Okay. But number five, this person, actually, he is the physicist, and he's the first person start to realize physics laws can be applied to the biology. Okay, so this is Schrödinger. So he developed a quantum theory, right? There's Schrödinger equation, quantum mechanics, and he has a book called "What Is Life?" Okay, and that at his time he started to think how to connect quantum physics with the life. Okay, so this book actually is very interesting to read as well. Okay, those are the great laws in the physics. Okay, and then you can summarize many laws, important laws in the physics field. Now, the question I asked is, does biology have the laws, theories, and equation? Okay, yes, they are. So first law actually is evolution. You guys answered that, raised that point. This is very good. Charles Darwin, right? He has the theory here. He said evolution, life evolved from simple form. One of the best tested, this is one of the best tested scientific theory, right? And the evolution is a series of tricks and random events to build upon it can evolution process can build a complex being from the simple parts. Okay, this is a theory. This is a correct biological theory. Second one is actually called the central dogma of biology. This is a theory fundamentally applicable either in the endothelial cell, muscle cell, neurons, fibroblast, all those cells. This central dogma theory applies. Okay, this is the law I'm talking about. It should be universal. Okay, now this central dogma talks about every cell has a DNA, and DNA can replicate itself, and then DNA can transfer information to the RNA. Okay, that process is a transcription, and then RNA can go to the protein. Okay, and then this process called translation. And then among here, DNA can replicate itself. DNA can replicate itself. Here, embed the genetic theory is embedded. I think some of you talk about the genetic theory, right? Discovered by the Mendel, right? So that is embedded here. Okay, but if that can be expanded to include RNA and protein. Okay, so here we will talk about in the next few lectures. Here, all the information here is actually linear. Okay, started from the linear form. DNA has the nucleic acid, RNA has nucleic acid, and then those nucleic acid actually is the alphabet of the life. And then when they connect together, they form the sentences, the words and the sentences. Every gene is a multiple nucleic acid, is a word. And then those words can connect into the sentences. And the sentences can connect into a paragraph. And then between those languages, they will connect it into multiple forms of information and then communicate it together to form the life. Okay, so this is another law. Third one, I want to talk about the equation. We talk about what is the equation in the biology, right? The one equation currently people find is on the membrane, on the cell membrane. So here is the cell membrane. Now, membrane is not that simple, actually. If you look at the membrane, you measure it. On the membrane, there are many transmembrane proteins. Those proteins can selectively segregate the composition of the ions to make them to be different inside the cell and outside the cell. And then, so those proteins actually select ions to let them to be distributed differently from between the inside and outside. So here actually is the cell. Due to those proteins on the membrane, actually the sodium concentration inside the cell actually is much lower than the sodium concentration outside. Okay, so inside is 5 to 15 millimolar, outside is 145 millimole. Okay, so this sodium is a salt, right? Sodium chloride is salt. Cells don't like salt, just like us. We need salt, but we don't eat a lot of salt. Okay, so sodium is different, and so on. Potassium is also different. Chloride is different. Calcium is different, and other ions also different. So you see this number, they're different. Now, this is just a chemical difference. These are ions, they carry charges. Those charges will be different inside and outside cell. And then those membrane actually is made of the lipid. Lipid is like the grease, right? Grease. They are isolated. If you learned the fundamental physics before, you remember capacitance, right? Parallel plate capacitance. Inside of the capacitance, there's a vacuum, but the plate is a conductor. Here, this is similar to a capacitor. Inside here is the grease, lipid. They are like the vacuum. They are non-conductive. But outside of the membrane, there's an extracellular region. They have a lot of charge. And the inside the cell, they have also a lot of charge. Those two different charges. Then this capacitance is overall charged, means they have a voltage there, have an electrical potential across the membrane, right? This can be calculated by an equation. So this is an equation. Okay. So based on the composition of the ion, we can calculate what's the membrane voltage there. Okay. This is not too surprise. It's not too surprise. What is a surprise is this membrane voltage can be perturbed. If you change it, this membrane will be similar to the will be similar to a circuit. The membrane will be looking at like a circuit. It will have a variable capacitance, variable resistance inside of it, because those ion channel, those protein can open, close, change their conformation. So the electrical signal on the membrane, electrical voltage on the membrane can vary. Okay, that is also not surprising. This is understandable. What is surprising is whenever this membrane start to change, they follow a unique and universal trend. So they go up first, go down second, and then become static. This trend is always the same for all the cells, as long as the cell can generate those perturbation. Okay, usually it's like a neuron, cardiac cell, muscle. They will generate this. This is called action potential. This signal is called action potential. So this membrane can generate a big pulse, and this pulse can propagate away. The equation I'm talking about is this equation. So this can be derived. This equation, okay? This equation not existing in any chemical engineering book or physics book. This is just because this biological system has unique transmembrane proteins, unique membrane properties. Then, by applying physical laws to understand the membrane, then you generate a new equation. This is a new equation in the biology. And then this one is called a Huxley model. Okay, and this is a Nobel Prize. So this is the equation in the biology. Okay. Oh, right now it's 3:57. We should stop here. So maybe we can discuss in the next time. Can you guys hear me?
Yeah.
Yep.
Excellent. Very good.
All right. Thank you. See you next week.
Thank you. Very good. You guys have very good